cancer. Oh. All right. So I'll just, yeah, you're already aware most of these things, kind of like, I'll be dealing with only adaptive immune system, forgetting about, for time being, innate immune system. Okay. So when you're talking about adaptive immune system, what are the important components? Okay. So the lymphocytes, then when you're talking about lymphocytes, B cells, T cells. Okay. So the B cells, the factories for antibodies, for making antibodies, plasma B cells. Okay. So there are plasma B cells and memory B cells. Memory B cells remember the pathogen. So when it comes second time, immediately makes antibody. While plasma B cells immediately makes antibodies for that particular infection at that point of time and try to tackle it. Okay. Then T cells are the important components. Among them, helper T cells and cytotoxic T cells. Helper T cells predominantly works in the context of external pathogens such as bacteria. But killer T cells or cytotoxic T cells, they take care of viral infections as well as cancer cells. That means they also take care of cancer cells. That means the cytotoxic T cells, they literally kills them. Okay. Then antibodies. Anyway, B cells. Then B cells produce antibodies, which kind of like tag the antigens derived from the pathogens or cancer cells. Then they try to neutralized by other components of the immune system. Then antigen presenting cells, so they take the pathogen inside. Then we also know that like antigen processing pathway, MHC1 and MHC2 for bacterial pathogens, MHC2 pathway, that means CD4 and this one T cells, CD4 T cells and MHC2 pathway, okay? So they recognize, the helper T cells recognize antigen presenting cells, presenting antigen. Then they try to kind of like gets activated and then activate the B cells. Okay, in that way, these antigen presenting cells are important to show the antigen to the immune components like such as T cells. Then major histocompatibility complex, MHC. Okay, so this is also kind of like CD4, CD8 and CD4. Okay, these are actually displace the antigens for the other T cells to recognize. Okay. Then cytokines, kind of like, these are the kind of like chemical messengers that will instruct B cells or other cells to kind of like make appropriate action. B cells mean to divide into plasma B cells and memory B cells, and the plasma B cells produce antibodies. Memory B cells remain intact and remember the pathogen. Okay. So then complement system, that is the kind of like ultimate final step of the all immune activity. That's what we'll want. So helper T cells, so usually kind of like the T helper cell recognize the same antigen presented by macrophages, which is APC, antigen presenting cell. Then it gets activated. Then again, when B cell shows or displays same antigen, then it recognizes that, activates B cell to become memory and plasma B cell. That means the B cell will be instructed to divide into plasma B cell and memory B cell. Plasma B cell produces antibodies, then it takes care of the antigen or the pathogen later. Then T cell. So the T cell also recognizes cytotoxic T cell or killer T cell, then infected tumor cell. Okay, so it latches with the various bindings. Then it releases granzyme and perforin, as Dr. Bapi was talking before. Granzyme and perforin. Perforin is the kind of like a bullet or something, you can consider that. It will go and create a pores on the membrane of the target cell, so that granzyme can enter inside the cell. The granzyme is nothing but a protease. Okay, that means it chooses or it fragments or it truncates or it cuts all the proteins being present indiscriminately in the target cell. So if it's cutting down all the proteins being present in the targeted cell, which are important for that cell to survive, that means it will be condemned to die. That means apoptosis. That means programmed cell death. So basically, the cytotoxic T cell or killer T cell sends granzyme and perforin to the target cell, maybe that's an infected tumor cell or cancer cell. So it can undergo programmed cell death or apoptosis or condemned to die. That's the basic mechanism. That's what it happens. So based on that, this is a fundamental knowledge of the immunology that people are trying to exploit. How can treatments can be devised or designed? Okay. So there are various immune checkpoint inhibitors. Okay. So these are nothing but like some of the molecules that are being expressed on cancer cells as well as immune cells, that is killer T cells. Okay. Say for example, PD1 and PDL1. PD1 is being present on the this one cytotoxic or killer cell, killer T cell. Okay. PDL1 may be present or will be present on the cancer cell. Okay. So the idea is that the cancer cell or immune cells are keep on like policing. That means they go around and look for bad things or bad cells and all. So whenever they come across something, they will try to bind to them. Okay. So in that sense, if cytotoxic T cell binds and also binds to this one, these two make contact, this PD1 and PDL1. PDL1 is the ligand. That means it binds to this one, present on killer T cell. Okay. So if they also make contact, it's kind of like a handshake. Okay. Oh, everything is fine. I'm not bad one. So then T cell thinks that, oh, it's normal cell, nothing to do with that. It goes off. Okay. It doesn't kill that. If PDL1 is not there, then if the cancer T cell binds to that killer T cell, then it understands it's a bad actor or bad cell, and it sends all this granzyme and this one kills it, literally. Okay. So when there is a handshake between these two, coming from killer T cell and the cancer cell, then nothing happens. Okay. So that means the cancer cell is, by intelligently expressing that one, trying to avoid its own killing when the police comes near to that. Okay. So that's what basically in layman terms. Okay. So now people, what they are doing is that they're trying to prevent this interaction, these two binding, when they come in contact. Okay. So those are called checkpoint inhibitors. That means they act as a brake so that the T cells can kill this one, the cancer cells. So how does it happen? So say for example, this is tumor cells. So PD1, this is a surface expression protein, surface protein. Okay. It binds to PDL1. Then there is a kind of like one more the antigen and the T cell receptor. So these also binds. So if this happens, nothing happens. If these two binding happens, so it's a kind of like a handshake. But if you are preventing this binding by creating anti-PD1, that means you design a molecule, drug, that goes and binds to that. So making, that means it cannot bind to anything now. Okay. So depriving opportunity for binding this. So now this T cell recognizes that one as a tumor cell, then sends granzyme and perforin so that it will be killed. So that is the kind of like active research that is being going on. So they found that like anti-PDL1 or anti-PD1 inhibitors, they designed that, there were clinical trials also, and it seemed to be very effective. Okay. So it's predominantly found for most of this kind of like liquid cancer, which seems to be the kind of like this strategy of drugs are very effective. Okay. Then there is a CAR T cell therapy. That means chimeric antigen receptor. Because T cells, T receptors are the ones that goes and binds and recognize the cells. Then after that, it makes a decision whether it's a good one or bad one. So if it's a bad one, then it will kill them. Okay. So that's what the strategy that they are going to use here. Instead of keeping intact T cell receptor, take it out from the patient itself. That means collect killer T cells from the patient itself, then modify engineer the T cell receptor. Okay. So they engineer in such a way that they put a new T cell receptor which recognizes the antigen or the surface protein that is unique to the cancer cell. Okay. So after engineering that T cell receptor, chimeric antigen receptor, CAR T, that's what it is. T means T cell, CAR means chimeric antigen receptor. Okay. So after putting that T cells again into the, that means they take it out the T cells, insert engineered T cell receptor, T receptor, new one, then they multiply that cells in the plate itself, in the lab itself. They grow them into millions of cells. Okay. Then they inject them into the patient. There won't be any incompatibility or kind of like immune issues because they are derived from the same patient. Okay. So then now after injecting, what they do, they will go and find the suitable, that means whatever the cancer cells having that particular antigen being expressed and kill them. Okay. That's what precisely happened in the context of CAR T cell therapy. Okay. So that's what happens. Remove blood from the patient to get T cells, especially killer T cells. Okay. Then make CAR T cells in the lab. That means first you insert gene that recognize that particular antigen being present on the cancer cell. Then again put it into the T cell, okay, which becomes CAR T cell. Grow into millions of these cells, okay. Then put back, infuse, inject literally into the patient, same patient, okay. Again they multiply there as well. Also they recognize cancer cells and kills them, okay. So that's in a very simplistic way of telling that, like how CAR T cells works, okay. Although it looks very nice, attractive and everything, it costs really a bomb. So much money, it's a kind of like, you have to literally kind of like take it out. Of course now I think the cost is kind of like being reduced because the mechanism is kind of like process is well established protocol. So, but even then, uh, patient-specific therapy. That means personalized medicine. That's what we call. That means for each patient, you are deriving a drug, medicine to treat. That means the CAR T cells designed for you may not work for somebody else. First thing is that these are derived from the same patient, so you cannot use it for other patient because of immune compatibility issues. Okay, so that's how these CAR T cells are designed for, are unique only for one particular patient. Okay, it works very well though, but it costs a lot of money because it's a highly personalized medicine. Okay. The cancer vaccines again, kind of like now people know that, like although we kind of like try to say that cancer cells also looks for all practical purposes like normal cells, but they do express something unique for them, unique surface proteins. Okay, so people try to identify them, then make cancer vaccines like how we make for COVID particular protein, capsid protein or something and all, right? So you kind of like, you can clone them, express them and inject, or you can also make mRNA vaccine. That means design mRNA for that particular protein which is important or which is being expressed on the cancer cell, inject it and try to develop vaccine. But these are, especially when you are talking about mRNA based cancer being pursued out, right, edge, okay, but something coming to the clinic or in the mRNA based vaccines done for cancer, probably they become, okay. So but there are some already available cancer vaccines are available, say for example cell for prostate cancer, that's kind of like well worked out and people are also using. Then some personalized neo-antigen vaccines which I talk about it. The monoclonal antibodies, these are actually, when you're talking about therapy for cancer, this is one already worked out thing. Say for example, triple HER2 breast cancer, HER2 rich cancer, there that protein is highly expressed monoclonal antibody that is trastuzumab, okay? That's available so that if you inject those subtype of breast cancer, it doesn't work for all types of breast cancer. One major subtype of breast cancer where precise targeted therapy is available. Okay. That's actually one of the major breakthrough. I'm not sure if it's been awarded Nobel Prize, but it's a very deserving candidate. Then cytokine therapy also being used for some of them, but not so effective, or I don't know whether it's really successful, and there are some instances, maybe possibly. Then even oncolytic viruses are also being tried. So the neoantigens, again, it's a very personalized medicine. That means even some people say it's well established that the cancer that you get is completely different from the cancer. Of course, although it's the same type. You are getting liver cancer. Your liver cancer is completely different from my liver cancer or somebody else's liver cancer. Okay, you cannot kind of like use same medicine to treat it effectively. Okay, in that context, these neoantigens are coming into the picture. That means each individual patient or that patient cancer cells exhibit particular type of antigens. That means proteins that undergo specific mutations. Okay, so what you do is that if that patient has to be developed treatment based on neoantigens, you collect that patient samples cancer cells, sequence them, all of them. That means like next generation sequencing or third generation sequencing, that next generation basically. So you sequence entire genome and try to find which proteins got mutated and which are being expressed in that patient. When you find that like a series of mutations or series of proteins, these are unique only for that patient, then you design antibodies for that. Okay? So that means it costs so much amount of money, few crores. Nobody can afford it. Only very rich people. But the thing is that like there is some sort of solution. Maybe over the period of time, it could be made affordable. We don't know. But people are trying this one. This is also one of the very cutting edge research area are actively being pursued to develop anti-cancer therapies for most of the cancers that is being tried, neoantigens. Thinking that like each individual patient has his own particular type of antigens for that cancer. Okay. So anyway, these two, CAR T cells and neoantigen comes as personalized therapy. That means each individual patient gets its own drug, basically. So the personalized neoantigen vaccines created based on tumor DNA sequence designed for that patient. They will not be useful for any other patient, only for that patient. Okay? So neoantigen specific T cell therapies, that means CAR T cells. Basically, you take out that own patient cells and engineer them, inject them, and try to kill the cancer cells. Okay? But that's what, as I said, kind of like there are many, again, kind of like they are not foolproof. You think that they are very specific, but we don't know. Kind of like they have many these kind of side effects as well, possible or anticipated, or some of them are proven that they may. And also that these personalized medicines are proven to be very good for treating or developing medicine for liquid cancers. The solid cancers, they are not shown to be very effective so far, because the solid cancer is an impact tight chunk of tissue. The penetration is the major important thing for any kind of treatment, right? The target should be accessible. If it's deeply buried inside somewhere, what is the point in kind of like sending something which cannot reach this one? But liquid cancers, they're free floating. Blood cancers, the cells are available, accessible in the bloodstream. So the drug that you design to target them can easily go and find them and target them. But when it comes to solid cancers, that it's that one major issue. Although you have the means and ways to develop particular type of therapy or treatments or drugs, but sending them to the correct place is a big problem. Okay. So again, not all patients do respond, delayed response. And again, as I said, that like tumor cells are so intelligent. I don't know why they are so intelligent or something, but they always become resistant for the period of time, though they may not respond. So that's it. Yeah. Any questions to ask? No questions. Did I make it clear? All right. Anyway, we can discuss informally as well. It's not that like in formal setting that you have to ask questions. No, not that way. All right. What is the time? The CRISPR-Cas9, yes, you can go, but thing is that how to penetrate them to all affected neurons, all the cells. There are millions of cells. So that's what called kind of like gene therapy. Right? Gene therapy means treating germline cells as well and somatic cells as well. So sending them that like kind of like to all the affected cells, that is the big task. It's possible that like